Hi everyone, today we shall discuss regarding glycosides. Okay, so coming to the definition, what is a glycoside? These are organic molecules derived from the plants and animals. So, these are complex organic molecules which can be obtained both from plants and animals, which on hydrolysis produces one or more sugar moiety along with non-sugar moiety. So it is made up of two parts. Okay, the glycosides inside them, they are made up of two parts. One is made up of sugar moiety and another one sugar moiety in the sense carbohydrates. Okay, so one is made up of a carbohydrate unit or sugar moiety, and another one a glycone part, that is non sugar moiety. Okay, so the sugar moiety is known as the glycone part, that is a carbohydrate product is present, and a non sugar moiety is known as a glycone part. So glycoside is a combination of sugar moiety, that is glycone, along with non sugar moiety, that is a glycone. Okay, so when you consider it as an example, glycone ROH, okay, plus a glycone non sugar moiety, right? So HOX, when you combine both of them, then you liberate out water. Okay, so these two combine and you liberate the water, and the remaining one is ROX, right, which is a glycoside. Understood, right? So that is about glycosides. Chemically, these are found as acetates or sugars or ethers, formed due to the interaction of alcohol or phenolic hydroxyl group. So how they are formed? These are found as acetates or sugars or ethers. How they are formed as acetates or sugars or ethers? Due to the interaction of alcohol or phenolic hydroxyl group. So when they interact with alcohol, or here with what it is been interacting, right? So when they interact with an alcohol or phenolic hydroxyl group, then they form into sugars or estates or ethers. So for example, when you consider here a carbohydrate, so we have taken a carbohydrate CH2OH, CHOH, 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 CH2OH. Okay, so when you are taking it and when you have interacted with an a glycone part, okay, that is CH3OH, when you have included that and you have interacted with that, then it liberates the water molecule, and this CH3O gets attached over here like this. Okay, here it got attached and the water is been liberated out. So this is methyl glycoside. Okay, it is methyl glycoside. So that is about the glycosides introduction. Next, color, colorless. Okay, exception cases, like some might be colored. So what are these? Anthracene glycosides are red in color, flavanol glycosides are yellow in color. So these two are exception cases. Next, when you go for the state, they are generally crystalline and amorphous in nature. They are crystalline and amorphous in nature. Next, solubility, they are water soluble and insoluble in organic solvents. Mostly have studied the same thing in all, right? Next, classification. So in general, these glycosides are been classified based upon two mechanisms. One is based upon the glycosidic linkage. So what is glycosidic linkage? With what a glycone part it is been linking with. Okay, so the link between a glycone and a aglycone, how they form a link, is known as the glycosidic linkage. Okay, so that is the glycosidic linkage, and based on chemical nature of the aglycone part. So and also based upon the chemical nature, so with what it is made up of, that chemical nature of aglycone part, not the glycone part, aglycone part. Okay, is these are the two mechanisms. So when we go for the chemical nature of the aglycone part, they have been classified into these categories, that is anthracene glycosides, sterol or cardiac glycosides, saponins, cyanogenetic glycosides, isothiocyanate glycosides, flavonoid glycosides, coumarin glycosides, aldehyde glycosides, phenol glycosides, steroidal glycosides, miscellaneous. So we have 11 categories in this. So you can also include um, you, you can also include examples for all these 11 categories. Okay, so like for example, if you take saponins, so here diosgenin, next ginsenoside are examples of saponins. Okay, this, and likewise you can write all the examples for all these things. That is about the chemical nature of the egg glycon part. Next, based upon the linkage, now we will go through the linkage. So how the links are been formed. So based upon that, we have C glycoside, O glycoside, N glycoside and S glycoside. So C glycoside, sugar moiety linked carbon atom, linked with carbon atom, sugar moiety linked with oxygen atom, sugar moiety linked with nitrogen atom, and the sugar moiety linked with sulfur atom. Okay, so when we go for carbon glycoside, also known as allione type of glycoside. Okay, allione type of glycoside, C glycoside. Okay, so what happens here, the glycone part. Okay, the glycone part ఇక్కడ చూసాం కదా, ROH plus HOX, glycone part, a glycone part. So here in the glycone part, so glycone OH. Okay, అక్కడ మనం ROH we have mentioned earlier as ROH, but here we are mentioning as glycone OH. Okay, plus HC. Okay, CH a glycone. So here we are having C linkage. Okay, so here water is been eliminating out and we are forming a glycone C a glycone, glycone C a glycone. This is the linkage. Okay, and water, very easy. This what we say, reactions are very easy to remember. Okay, so examples are anthracene glycosides, example cascara, example cascara, anthracene glycosides, example cascara, C glycoside, cascara. Okay, and here another type what we studied, it is also known as alloyne type of glycoside. It is mainly for Liliaceae family, example aloe. ఓకే, ఈ టైప్ ఆఫ్ మనం మెన్షన్ చేస్తే వీ నీడ్ టు రైట్ ఎలోయిన్ మెయిన్లీ ఫర్ లిలియేసీ ఫ్యామిలీ. ఓకే, ఎగ్జాంపుల్ ఎలో. Okay, that is about C glycosides. Next, O glycosides, example senna rhubarb. So for senna rhubarb, here see, glycone plus OH, next CH glycone అనుకున్నాం కదా. సో ఇక్కడ ఏమొస్తుంది, వెరీ ఈజీ నో, OH. ఓకే, glycone OH plus OH glycone. Okay, water is liberated out, glycone O glycone, right, the formation of O linkage. Okay, so here examples senna rhubarb. Next, S glycosides, examples isothiocyanate, signin from black mustard. In the category of isothiocyanate, cynigin. Okay, cynigin from black mustard. Okay, that is the example. And you know the reaction like glycone OH plus, okay, SH glycone. So H2O will be liberated out, you will get glycone S glycone plus H2O. Okay, so those are the examples, and same with the case of N glycoside also. N glycoside also you will have the same linkage formation. So those are the types of glycosides based upon the linkage. Okay, glycosidic linkage. Next, the chemical portion, or the chemical property, or the physical property, or the therapeutic property of these glycosides are mainly responsible because of the egg glycone part only. Okay, whatever chemical reactions or physical properties they are showing, or therapeutic activity they are showing, they mainly depend upon the egg glycone part. Next, coming to the chemical tests. Okay, when we go for the chemical tests, how they are been chemically tested. So we have the Killer-Kilani test. Killer-Kilani test. Sample plus 70% alcohol is been taken and it is been boiled for 3 minutes. We take the sample and we mix the alcohol and we boil it for 3 minutes and then we filter it. Then what we will get? You will get a filtrate, right? To the filtrate, water is added and lead acetate is added. Okay, and you completely shake it and then you again filter it, and then you'll get a clear filtrate. Okay, transparent clear filtrate అనేది ఫామ్ అవుతుంది అనమాట. To that filtrate, suppose it is of 10 ml. Okay, that filtrate is of 10 ml. So same 10 ml of chloroform, equal volumes of chloroform has to be added. ఓకే, సో ఇది 2 ml లేకపోతే 3 ml ఉంటే అంత ఫిల్టరేట్కే మనం క్లోరోఫామ్ కూడా యాడ్ చేస్తాం. సేమ్ అమౌంట్ క్లోరోఫామ్ యాడ్ చేస్తాం. ఆ 2 ml 3 ml క్లోరోఫామే యాడ్ చేస్తాం. ఓకే, దాని తర్వాత వి గో ఫర్ ఇవాపరేషన్. సో వి ఇవాపరేట్ ఇట్. దెన్ యూ ఎక్స్ట్రాక్ట్ విత్ గ్లేషియల్ ఎసిటిక్ యాసిడ్ అండ్ యాడ్ ఫ్యూ డ్రాప్స్ ఆఫ్ ఫెర్రిక్ క్లోరైడ్. Okay, you add glacial acetic acid and few drops of ferric chloride. Approx solution form అవుతుంది కదా. Then to that you add H2SO4 from the walls of the test tube. Okay, from the walls of the test tube you add H2SO4, sulfuric acid యాడ్ చేస్తాం. Then what happens, the whole solution ఇలా డివైడ్ అయిపోతుంది అనమాట. Okay, and at the junction of both the layers you will get a reddish brown color ring. See, like this. Okay, you have already done this somewhere, like or similar type of experiment you have done. So you will get a junction with reddish brown color ring. Whereas the upper portion what is present, no, that appears to be like a dark bluish green color. It shows a dark bluish green color. Below that you will get a brownish color ring, and below one will be transparent. H2SO4 యాడ్ చేసే కొలది ఏమవుతుందంటే ఆ లేయర్ అంతటిని డార్క్ కలర్ లేయర్ అంతటిని పైకి పంపిస్తుంది. H2SO4 కింద ఉంటుంది. ఆ మధ్యలో ఏమొచ్చి యూ విల్ గెట్ ద బ్రౌన్ కలర్డ్ రింగ్ అనమాట. ఆ రియాక్షన్స్ అన్నీ జరుగుతాయి. ఓకే, ఆర్ రియాక్షన్స్ జరిగి గ్లేషియల్ ఎసిటిక్ యాసిడ్ అనేది కిందకు వచ్చి ఆ బ్రౌన్ కలర్ రింగ్ ఫామ్ అయ్యి యూ విల్ హ్యావ్ ద అప్పర్ పోర్షన్ విత్ దిస్ డార్క్ బ్లూయిష్ గ్రీన్ కలర్. Okay, so this dark bluish green color indicates the presence of cardiac glycosides. మనం తీసుకున్న శాంపుల్లో ఇఫ్ వీ హ్యావ్ కార్డియాక్ గ్లైకోసైడ్స్ దెన్ ఇట్ ప్రొడ్యూసెస్ ద డార్క్ బ్లూయిష్ గ్రీన్ కలర్. Okay, on the upper portion this ring appears to be brown in color. Okay, so that is about the Killer-Kilani test, so which is for glycosides. Remember properly, Killer-Kilani test which is for glycosides. For tannins we have studied the gold beater test, right? For glycosides we have studied the Killer-Kilani test. We also have another test, that is Bulgett test. Okay, Bulgett test. Here what we are doing, here we are taking a section of the drug like transfer section. Okay, we are taking the transfer section of the drug plus sodium picrate solution. Okay, then we are taking sodium picrate solution, and when we mix them there will be a color change from yellow to orange. Okay, there will be color change from yellow to orange. That is Bulgett test. Section cutting తీసుకుంటాం, తీసుకున్న దాంట్లో sodium picrate solution అనేది యాడ్ చేసేస్తాం. Then we will get a color change from yellow to orange, which indicates the presence of glycosides. Okay, so when we go for uses, so what are the uses of glycosides, which is the most essential thing which we need to study, right? So when we go for that, we have the hyperglycemia, example Mamadika charantia, which has been kakargai. Okay, which has been mostly used for the diabetes in Ayurvedic preparations and everything. So we have hyperglycemia, example Mamadika. Diuretic we have erythrogenin, which has been obtained from gokru. Okay, gokru. Next, antidepressants we have hypericum. Okay, next vitiligo we have xanthotoxin. Vitiligo like blackening of the teeth, for that we use the xanthotoxin. Okay, next hepatic diseases we have silimarin. So these are few of the uses by which the glycosides which are present in these all things, okay, all these drugs are helpful for reducing these diseases. So that is about your glycosides topic. Very easy topic. If you once read it, you will get it. Okay.